So, what do we mean by phylogenetic comparative methods? Um, we use this term to refer to analysis where we're asking comparative questions about some data sets. We're trying to test some hypotheses, but we want to account for phylogenetic relatedness between our data points, which often are groups or species or populations. So if we can show an example here, imagine that you're interested in the relationship between some variable x and some other variable y. You have collected data on x and y, and you would like to know if there is a significant relationship. So these could be characteristics. Ecological characteristics, for example, except that your data points here are not individuals from a given population, for example, they are different species or different groups. And that is important to realize because these species or these groups may have a phylogenetic history. Actually, they do have a phylogenetic history. And that is what we symbolize with this tree like underlying structure here. So some points may be more related than others. And what that means is that these data points are not necessarily statistically independent from each other. And that is a big deal because in statistics, when we test hypotheses in this kind of framework, especially in using linear regressions, for example, uh, we assume that the points are statistically independent. So what does that mean? Uh, the fact that groups of points are not statistically independent from each other mean that they might be pseudo replicates of each other. And pseudo replication basically means that you may have, if multiple points are actually uh, very non-statistically independent, they might basically be equivalent to just one single point. And in a phylogenetic context, this would correspond, for example, to three species that are very, very highly related, closely related. So, for example, the three species in the top right of this plot are relatively close together according to the phylogeny. They might be pseudo-replicates of each other. So they might not constitute three independent data points to test our hypothesis. They might be just pseudo-replicates of one single data point, just because they're so closely related to each other. In other words, if we forget about the phylogeny and we just look at the cloud of points, we have the impression, and that's what the green line, the regression line, is telling us. We have the impression that there is a relationship between y and x, but if the 3 points on the top right are actually not statistically independent and kind of are uh, each the kind of each representative of one same point, that would mean that in this top right corner of the graph, we only have 1 point, not really three, because they all represent, all are slightly different versions of the same one because they are so closely related phylogenetically. If we only had 1 point there, then this pattern of increase that we have identified would be much less convincing because it would very much look like we only have one outlier that happens to have a high x and a high y, but it would not be a very convincing evidence that indeed variable y increases with variable x. And so this is why it's important to consider phylogenetic relatedness when we work with this kind of comparative studies, because we may conclude stuff just by looking at clouds of points. We may conclude that there are some patterns exist, while actually they don't really, if we do take into account that uh, data points are not completely independent from each other. So we may inflate the impression that there is actually a pattern where there might be just nothing. So how do we do this properly? Where there are different methods that people have come up with. The two main ones are first this method of phylogenetically independent contrast that Joe Felsenstein came up with. Basically, I'm not going to go into detail here. Uh, The idea is to identify pairs of points of pairs of nodes in this phylogenetic tree that we want to care about, in order to reduce the biases due to this non-independence. And another method is called phylogenetic generalized least squares. So it's like an improvement on some statistical methods for linear regression that basically says, well, you have to assume that the different species that you're studying along some axis, so x or y, or one of these two variables, these variables have evolved along the branches of this phylogeny. You have to assume a certain model, a mathematical model, according to which this could have evolved. So usually people use very simple mathematical models. One very simple one is called the Brownian motion model, where we just assume that this trait, this variable x, has evolved along the branches of the phylogeny in a random walk kind of fashion, like the diffusion of a gas, basically. But there are other models that can be used. And basically, this assumption that a model has generated the spread of these variable in the is taken in the background, basically, when this linear regression is being fitted with these phylogenetic generalized least squares. And the idea is this statistical procedure is going to account for shared ancestry in fitting this statistical model. So it's basically just explicitly accounting for the fact that some species might have related values of x and y, uh, not independently, just because they share an ancestor relatively recently in the past. And then say, well, taking that into account, now do we still detect a positive relationship, for example, this green line between our two variables, after we have accounted for phylogenetic relatedness? Now, in practice, there's not always a clear signal that there is a strong phylogenetic there's a strong phylogenetic signal that should be accounted for, but usually it's nice to there are packages and techniques to basically take this into account. So it's always a good idea when you're working with comparative data sets with multiple species, for example, to think about checking for these phylogenetic signals. And then if the method you're using is telling you, yes, there is a phylogenetic signal, then it's good to use one of these methods to account for it.